So again, by the end of this class, you will know a secret code that most people don't. And it is a secret code that works for every living thing we've ever seen. I don't want to oversell it, but it's like almost like the secret of just reminder, this is, no, don't write this. You've already done this. This is Thursdays. The transcription is part 1 of gene expression, and you've done that on paper, showing that the RNA polymerase unzips the DNA and adds complementary RNA nucleotides at the same time. Once that strip of mRNA is complete, it peels off and kind of leaves the DNA to zip back up on its own. And we know it's going to leave the nucleus. We modeled what it does out of the nucleus with the paper shapes on Wednesday, anyway, to where the tRNA grabs amino acids and brings it in. Before we get into that, though, something still has to happen to this thing before it leaves the nucleus, something we haven't talked about. What happens is it turns out there's some junk. There's some stuff in that messenger RNA that is not necessary to make the protein. And so what happens is a big old protein complex comes in and edits out the junk parts. For our purposes, we'll say the middle part is the junk part. And it splices those parts back together. Yes, Hazen, you should be writing this. Okay. Yep. And we are left with what is called the edited mRNA transcript. That ending looks different depending on which gene we're talking about. There's no real way to predict, at least for us, to predict which will be the things that get cut and which things will actually get spliced together to make the real protein. But we know it happens. I think the challenging thing about this terminology, at least for me, it's one of those where the terms kind of seem backwards to me, introns and exons. Yeah. Yeah. So that's one way to remember it, actually, is that it's just the reverse of what you think it's going to be. Exons are the good parts, introns are the bad parts. The other way, the way I usually remember it, is that the introns, since they're junk, they have to stay in the nucleus. They never make it out of the nucleus to make the protein. The exons get to exit the nucleus and go do the work. Whatever works for you, but you need to be able to sort out introns from exons. And the next thing we look at, translation, there's going to be a bunch of parts kind of moving and bringing stuff in and dropping stuff off and hooking stuff together like you did with the paper shapes, but kind of cooler. But before we even get to that, we kind of get to cheat. We're going to skip to the good part. Do you still get prizes in cereal boxes? Man, I'm sorry, because that was one of the best things.
Like as children, we got them, but now.
And I think it even by your time had changed to like it wasn't actually in your food. It was like outside the bag.
Yeah.
Man, when I was a kid, it was like I had to dig my little chubby arm all the way down into the Honey Nut Cheerios and like root around in there and find a little thing. Or sometimes I just dump, like I think a couple of times I got yelled at for just dumping out the cereal and grabbing the prize. Anyway. Yeah, I usually had a little cellophane wrap thing. But I mean, the point is that there was like toddler arm in the, you know. Anyway, some of those toys were cool and some were not. One of the ones I liked was the secret decoder wheel. You know what I'm talking about? Like it was usually just a simple like alphabet substitution code. You spin the wheel and it, okay, A is G and B is H and so on. Yes. Like the one from Christmas Story. Right? So I don't know if that's the same one, but yeah. Come on. Wow. Because here's the thing. Oh. We know the code. I'm gonna take you through, on either tomorrow or Wednesday, and take you through the machinery and the tRNA and the ribosome and all that. But we don't have to do that right now. We can just skip to the code part. We know which mRNA bases code for which amino acids to make proteins. And the code isn't that complicated. It's a 3-letter code, basically. So you just have to look at the edited mRNA, 3 bases by 3 bases, basically little triplets. And we know exactly what those 3-letter sequences code for. And I'm going to give you that code. I am going to give you your very own, for keepsies, secret decoder wheels. I know, I'm the best. I'm going to teach you how to use it. It's not hard. Some of you will figure out how to use it by the time I get done passing these up. The amino acids, that is the things that make up proteins, there's 20 of them. And they are listed around the outer ring of your secret decoder wheel. If I was a better teacher, I would have laminated them for you. But there you go. Look, can you tell what you do to get to the amino acids? It's fairly intuitive. Where do you start? Yeah, you start with the big, huge letters in the middle. There's only 4 of them, right? So if we want to know what AUG codes for, AUG, that's the first, we call these codons, by the way. The first codon is AUG. We start in the middle and we find the A, the biggest A there is, right? This A. That means that we are now in this lower, call it the southwest quadrant, right, of the wheel. So we're stuck there now. And then we go to the second letter, which is U. Well, now we go 1 ring out to the medium-sized letters. And we see that U is basically the space between 6 and 7, if this is on a clock, right? 6 and 7 o'clock. And so we're stuck in the U now, and now we hit the G, which is in the smallest second to outermost ring. And that restricts us to just 1 amino acid name, and that is methionine, is how we say that. And methionine turns out to be the most common first amino acid in any protein. If you see methionine as the first one, you're probably doing the decoding correctly. And that's literally all there is to it. Well, okay, not quite, but close. You use your secret decoder wheel, and real quick in the margins of your notes, write down what the other 2 amino acids this codes for. In other words, we did AUG together. Give me what CUC and AAG code for. Is that easy? So CUC, we're in that, let's see. We're in the southeast quadrant now, right? Which amino acid did you wind up with? Great. C, and then U gives you about 330, and then C, yeah, leucine. And you notice with leucine, this is one of the details you need. What if there's a mutation that changes that 3rd letter? What amino acid does that change it to? Doesn't change it, does it? That 3rd letter actually doesn't matter on leucine. It can be any of the 4, U, C, A, or G. And you can see there's other amino acids like that with that. We call that the wobble base, where there's that 3rd base doesn't matter much. If you get the first 2 right, you're there. And we'll talk about mutations soon and why that matters. Good. And then the next one, the 3rd one, AAG, codes for what? Armani. Yeah, lysine is how you say it, AAG, and there's 2. If that was a G or an A, it would be the same thing. That's lysine. Good. Yeah, that's kind of all there is to it, except look around the ring. It's all amino acid names. All 20 amino acid names are there. But then there's another thing that you might notice that might, yeah. There's three spots, UAA, UAG, and UGA. Instead of an amino acid name, they all say stop. Guess what that is? Say it again.
Do they not have a base maybe?
You're right that doesn't attract an amino acid. Huh? Telegram? Is that what you said?
If you put those together, will it make something bad?
No, well, if it's in the wrong place. Chris? Huh? It does not make things explode.
Does it just make excess of something that's the body?
Is it like a flaw in the gene? Like
Not usually. Usually it's right where it's supposed to be. That's how you know the protein's done. A gene is a code for a protein, right? So you got all these letters that code for all these amino acids. Well, the thing has to know when it's done. And so at the end of the code is one of those 3 codons that says stop. And in fact, instead of attracting an amino acid, it attracts a release factor that's basically a pair of scissors that cuts the protein off and lets it go. And if it's done right, it's a functional protein. That's it. That's all there is to it. So today's assignment, since we don't have a lot of time today, I'm just going to put you right into decoder mode. I'm giving you a real protein to decode. I'm giving you the edited mRNA transcript for insulin. Now, the human insulin gene is a little longer than you would want to deal with. So this is a cow insulin protein. Insulin, of course, is the protein that you release that is a hormone that lowers your blood sugar. It's all about blood sugar. It's a hormone made in your pancreas, also made in the cow's pancreas. So you're going to need your decoder wheel. And here's the deal. Take a look. You see that the little bubbles are numbered, right? And in fact, we've taken the liberty of doing the first 4 amino acids for you. 1, 2, 3, and 4. You can see the first 3 letters of the amino acid are fine. That's how we're going to do this. So you write the first 3 letters of the amino acid in those little bubbles. And I want you to do 1, 2, 3, and 4, which, again, the mRNA is up here. Right? It's this big, long chain of letters. We've kind of subdivided them into 3s for you, into codons. Go through with your wheel and check yourself. For those first 4, you should get the same answer we did. If you do, then you know you're on the right track. And it should be easy from there to go ahead and just go 1 through 30 on the protein, and then skip back up to 31, which is the second chain. Okay, stop looking for a second. Look up here, because there's more. So that part you know now. Then there's 2 questions. Answer those as best you can. Then number 3, you're going to be like, oh, is he joking? He is not joking. I do want you to write the DNA from which this mRNA would have come from. In other words, I want you to go through and write all the letters that would have matched up with UUU, CUC, and so on. It's a lot of letters, but it's really good practice for me to know that you're good at this whole complementary base pairing thing. What will the first 3 letters be, by the way? What in DNA would match up with U in RNA? Yeah, see, you need to practice. What is it? A. So A. Go ahead and write down A under number 3. That is the first 3 DNA bases. Good? That is your homework. That homework is due tomorrow if we are here. It is due Wednesday if we are not. So I'd say pretend like it's due. Yes, I think that it'll be just like, you know, as soon as they get those documents today, if they do, we'll all get zapped with little text.